Very excited and happy to have the talented Shani Peleg. She's an MD PhD candidate working in the lab of Yuval Dor. And the talk title actually, sorry, is RNA editing deficiency models, differential immunogenicity of alpha and beta cells. And a short bio for Shani is, again, she's an MD PhD student at the Hebrew University in Jerusalem. She completed her PhD investigating the role of RNA editing in pancreatic endocrine cells under the supervision of Professor Yuval Dor and Dr. Agnes Chandler. I'm going to ask people to drop questions in the chat. We'll take them at the end. And here also in the chat is the link to the newest paper. Thank you, Shani, so much for joining us. And we're really excited to hear more from your talk.
Thank you, Monica. Thank you so much for this opportunity to present here. My name is Shani. I'm from Yuval Dor's lab. And today I'll be presenting findings from our recently published paper in Molecular Metabolism on our study of RNA editing deficiency in pancreatic endocrine cells, which models differential immunogenicity of alpha cells and beta cells. So as we all know, type 1 diabetes is an autoimmune disease that's characterized by selective destruction of pancreatic beta cells. This is the classical model that's used to describe the pathogenesis of type 1 diabetes. So in the context of genetic predispositions, it's thought that there's a triggering event that induces an autoimmune process, leads to beta cell death, which leads eventually to clinical onset of type 1 diabetes. Now, how this autoimmune response begins is unknown and is a major question in type 1 diabetes research. But we do have a hint that may explain how this all begins, and that is that the autoimmune process that precedes type 1 diabetes is a interferon response. So this autoimmune process is preceded by a type 1 interferon response. So what is a type 1 interferon response? This is a response that's elicited by microbial products, and its aim is to begin antiviral cellular processes that will eventually resolve infections. This occurs through the stimulation of pattern recognition receptors, such as Th1, by different microbial products, such as double-stranded RNA. And this sensing begins signaling pathways that end in the production of type 1 interferons, interferon alpha and beta. And these type 1 interferons bind to JAK-STAT to interferon receptors that activate the JAK-STAT signaling pathway. And this leads to the expression of interferon-stimulated genes and begins different antiviral activities. So there's accumulating evidence that shows that there's an important role for the type 1 interferon response that precedes type 1 diabetes. So, for example, recent onset patients have elevated type 1 interferon signature in their blood and in their islets. In addition, GWAS studies have found an association between polymorphisms in Th1, which is a double-stranded RNA sensor that activates the type 1 interferon response, to type 1 diabetes. So they found that gain-of-function polymorphisms in Th1 are associated with increased susceptibility to type 1 diabetes, and loss-of-function mutations are protective for type 1 diabetes. Now, what could cause this type 1 interferon response that happens before the onset of type 1 diabetes? The prevailing theory has been the viral hypothesis, which would make sense, right? A viral infection could lead to double-stranded RNA sensing by Th1, which would lead to a type 1 interferon response, islet inflammation, and maybe type 1 diabetes. And the viral hypothesis has been studied profusely over the years, but a definitive causal relationship between viral infection and type 1 diabetes has still been lacking. So we asked ourselves whether there could be a viral-free explanation for the double-stranded RNA-mediated type 1 interferon response that precedes type 1 diabetes. And this is how we began to study deficient RNA editing in pancreatic endocrine cells. So what is RNA editing? RNA editing is a fundamental cellular process that's mainly conducted by adenosine deaminases acting on RNA, ADAR enzymes. ADAR deaminates adenosine bases into inosine bases, which are read by the translational machinery as guanosines. Vertebrates have 3 ADAR proteins, ADAR 1, 2, and 3. And today we'll be focusing on ADAR 1, which is responsible for the majority of editing activity and is ubiquitously expressed. Now, the most essential role of A to I RNA editing by ADAR 1 was thought to be in recoding of the resulting protein, but recent studies have revealed that the main role of ADAR 1 is to disrupt endogenous double-stranded RNA structures. So these double-stranded RNA structures are formed mainly by retroelements that are inserted in an inverted orientation in our genome, mainly in the 3' UTR of transcripts. So these are ALU-ALU inverted repeats in humans and B1, B2 inverted repeats in mice. Now, when these inverted repeats are transcribed, they form endogenous double-stranded RNA structures, which could be mistaken for viral double-stranded RNA. And this could lead to an antiviral interferon response, even though no such response is needed. And this is where ADAR comes in. So ADAR destabilizes the endogenous double-stranded RNA structure and prevents an unnecessary immune response. So in our work, we propose an alternative model that could explain the interferon response that precedes type 1 diabetes. Maybe the origin of this double-stranded RNA isn't viral, but endogenous, which could result from lower levels of RNA editing. This would then lead to the accumulation of double-stranded RNA, the sensing of the double-stranded RNA by the double-stranded RNA sensor Th1, inducing interferon signaling and innate immune response, and maybe under the wrong genetic background, this could lead to type 1 diabetes. And really, our model was supported by a paper that was published by Jin-Billy Lee's team in 2022 that shows that deficient RNA editing might underlie genetic risk of different inflammatory diseases, including type 1 diabetes. So briefly, they first found genetic variants that are associated with decreased levels of RNA editing. They then found that there's a significant overlap between genetic variants that are associated with decreased levels of RNA editing and genetic variants that are associated with autoimmune diseases, such as lupus, psoriasis, IBD, and also type 1 diabetes. So this paper supports our hypothesis that impaired RNA editing in islets may induce the interferon response that precedes type 1 diabetes. And in order to study this hypothesis, we developed a mouse model with inducible deletion of ADAR in beta cells, with GFP as a reporter for mutant cells. So upon injection of tamoxifen, we induce the deletion of ADAR, specifically in beta cells. These are our beta ADAR knockout mice. And these results in the next slide have been conducted mainly by Udi from our lab and have been published in Cell Metabolism last year. So what happens when we delete ADAR in beta cells? This caused a dramatic phenotype. So first of all, ADAR knockout caused diabetes, which we can see here in the random glucose of knockout mice in green versus controls in yellow. And this was supported by a decrease in beta cell mass in diabetic mice that are beta ADAR knockout. And what leads to beta cell death in our beta ADAR knockout mice? Massive islet inflammation, which peaked at around 12 days post-tamoxifen injection and was heterogeneous in nature. So we see here islets with GFP in green, CD45, which is a panleukocyte marker in white, and insulin in red. And we see that there are some islets that have minor inflammation, while other islets have severe inflammation. And this inflammatory phenotype was supported by an elevated interferon response in beta cells of beta ADAR knockout mice, which we can see here in green. This is a panel of interferon-stimulated genes, and we see an induction of interferon response in mutant beta cells. And the third thing that we saw was decreased insulin expression in inflamed islets of beta ADAR knockout mice. So there's an inflamed islet here of beta ADAR knockout mouse, and we see decreased insulin staining in the same islet. So ADAR deletion in beta cells had a very dramatic response with detrimental effects to beta cells. And what was interesting was that mice with Adar loss of beta cells had many similar features to type 1 diabetes. So we have beta cell dysfunction and destruction, heterogeneous islet inflammation. We also saw age-attenuated severity, so that younger mice presented with more severe phenotype. And we found that if we increased beta cell metabolic load, this also led to a more severe phenotype. Lastly, and this is what I'll focus about in the rest of the presentation, was that we found that in contrast to the detrimental effects on beta cells, other islet residing cells were spared in this model. So indeed, very surprisingly, Adar loss in beta cells doesn't seem to affect neighboring alpha cells. We can see here again that inflamed islets of beta Adar knockout mice with inflammatory cells here in red, we see that insulin staining is decreased. In contrast, glucagon staining in inflamed islets remains unchanged. And in even more extreme cases, we saw that islets were composed suddenly of a majority of alpha cells, when usually in mouse islets, alpha cells make up around 15 to 30% of the islet. And this phenomenon is reminiscent of an elusive phenomenon that occurs in type 1 diabetes, where we often see alpha cell persistence. And really, this is a major question in type 1 diabetes research. Why are beta cells and not alpha cells targeted by the immune system? Do alpha cells have some sort of innate protection against inflammation? So we decided to use our model of deficient RNA editing to dive in a little deeper into these fascinating differences between alpha cells and beta cells. So we decided to use a second mouse model, this time with inducible Adar deletion in mouse alpha cells. Again, with GFP as a marker of mutant cells. So these are our alpha Adar knockout mice. We wondered whether we would see similar effects on alpha cell function, alpha cell viability, and immune response, as we saw in our beta Adar knockout model. So we began by investigating how Adar knockout in alpha cells affects alpha cell function. We performed many different physiological tests to do this, but we found that impaired RNA editing in alpha cells doesn't affect alpha cell function. And this is in contrast to the detrimental effects that Adar deficiency had on beta cells. And what about inflammation? We were very surprised to see that mouse islets with Adar deficient alpha cells did not attract immune cells. We can see here islets stained for GFP in green, glucagon in red, and CD45, the pan-leukocyte marker, in white. And we checked for inflammation upon many different time points, but we didn't see any inflammation at any time point. And these results were reflected by the interferon response that we saw in mutant alpha cells. So here we can see the expression of a panel of interferon-stimulated genes in beta Adar knockout beta cells and alpha Adar knockout alpha cells and their controls. And we found that the interferon response in Adar knockout beta cells was significantly higher than the interferon response induced in Adar knockout alpha cells. So we conclude that impaired RNA editing in alpha cells doesn't impair alpha cell function and leads to only a minor interferon response, which is not enough to attract immune cells. So far, we saw that in our beta Adar knockout model, we saw islet inflammation and interferon response that do not impair alpha cell viability. In our alpha Adar knockout model, we saw that cell-autonomous RNA editing deficiency in alpha cells also doesn't impair alpha cell viability. So we asked, what would happen if we combined the two? And to do this, we used a third mouse model with Adar knockout in both alpha cells and beta cells, our Adar double knockout mice. And we wondered whether a combination of an immune attack that would be triggered by beta cell Adar deletion combined with cell-autonomous processes that would be induced by alpha cell Adar deletion, if this would finally lead to alpha cell death. And indeed it did. So we can see here islets from our 3 mouse models, stained for glucagon in white, insulin in red, and somatostatin in green. And we can see that in the alpha Adar knockout mice, we have normal islet architecture, normal insulin and glucagon staining. In the beta ADAR knockout mice, we suddenly see a significant decrease of beta cells or insulin-positive stain cells in islets. And in our ADAR double knockout mice, we see a decrease in both insulin and glucagon. And we quantified this, we quantified the alpha cell area, and we found that there's a significantly decreased alpha cell area in ADAR double knockout mice. So there is alpha cell death when we combine islet inflammation and ADAR deficiency in alpha cells. So to conclude this part of the presentation, we found that alpha cells are resistant to the impacts of RNA editing deficiency, in contrast to beta cells. So we saw here very little interferon response, no inflammation, and no effect on alpha cell function or viability. In contrast, beta cells are very sensitive to RNA editing deficiency. We see an augmented interferon response, islet inflammation, impaired beta cell function, and beta cell death. Now, another important finding that we had in this model was that in these beta ADAR knockout mice, wild-type beta cells were also targeted. So mutant beta cells weren't the only ones targeted. We also saw wild-type beta cell death, and it was in the same amount or the same ratio as beta cells were, as mutant beta cells were targeted. So it looks like wild-type beta cells are more sensitive than wild-type alpha cells to death from inflammatory cytokines that are produced in this model. Lastly, we found that alpha cell death can be induced if we delete ADAR in both alpha and beta cells. So after discovering these interesting differences between alpha cells and beta cells and their sensitivity to RNA editing deficiency, we were left with many questions, and the big one being, what is the mechanism that underlies this phenomenon? And we began to answer this in our paper, so I'll present our findings briefly. We focus on the signaling pathway that happens when ADAR is deleted, and we try to isolate which steps in this pathway might be different between alpha and beta cells, and what step might explain this differential sensitivity to RNA editing deficiency. So first of all, ADAR knockout in alpha cells was as efficient as it was in beta cells. So there's no difference in knockout efficiency between the cell types, between our models. Second, previous findings from Desio-Isrich's group have shown that alpha cells are as sensitive to double-stranded RNA as beta cells are. So it's not this part of the pathway either. Third, alpha cells don't have lower basal expression of double-stranded RNA sensors, such as Th1 or other interference-mediated genes. So it's not this part either. And fourth, alpha cells are as sensitive to type 1 interferons as beta cells are. So up till now, I presented our main findings that we've published in Molecular Metabolism this year. And regarding the mechanism, we have managed to narrow it down, but we haven't reached a final validated mechanism that underlies this differential sensitivity of alpha cells and beta cells to ADAR knockout. And we currently have two leading hypotheses that we've been working on at the lab. So our first hypothesis, so if we look at this signaling pathway that we've investigated, there is one step that we haven't yet investigated. And that is, what if the differential sensitivity of beta cells and alpha cells to ADAR knockout lies within the types of the double-stranded RNA that each cell transcribes? What if beta cells produce more dangerous double-stranded RNA, either more in quantity or maybe specific double-stranded RNAs that are more immunogenic? And maybe the transcriptomic landscape of alpha cells is less immunogenic than the one of beta cells. And what could maybe cause beta cells to transcribe more immunogenic double-stranded RNAs? This could possibly be related to metabolic load. And this idea comes from findings from our previous cell metabolism paper. We showed there that increased metabolic load on ADAR-deficient beta cells led to an augmented interferon response. And we think that by increasing the metabolic load, we are in effect changing the transcriptional landscape of beta cells. And we might be inducing the transcription of more dangerous double-stranded RNAs. Again, either more in quantity or simply specific double-stranded RNAs that are more immunogenic. So our hypothesis is that if we increase the metabolic load of alpha cells, we might be able to trigger an augmented interferon response that maybe resembles the one that we see in mutant beta cells. Our second hypothesis is that maybe these differences between alpha cells and beta cells is related to islet architecture. And we've begun to investigate this direction. And in the last few minutes, I'd like to share some exciting unpublished preliminary data. So for this hypothesis, it's important to understand that the interferon response is dependent upon a positive feedback loop. So we have here an ADAR deficient cell, and it will begin to secrete type 1 interferons. These type 1 interferons will bind to other cells in the area. And if the cell is ADAR deficient, it will produce more type 1 interferons, which will bind to more receptors, which will lead to more expression and secretion of type 1 interferons, et cetera, et cetera. Now, what happens when this mutant cell is located in proximity to other mutant cells? The type 1 interferon that's secreted by these adjacent cells will bind to other mutant cells, leading to more and more type 1 interferon secretion, an augmentation of the interferon response. And this could later attract immune cells. Now, in mouse islets, alpha cells are located in the perimeter of the islet, while beta cells are located in the core. So in beta ADAR knockout islets, we have many mutant cells that are in close proximity to each other. And in contrast, in alpha ADAR knockout islets, the mutant cells are more dispersed. They're further away from each other. So we could imagine that there's less communication between these cells and maybe therefore less positive feedback loops and less augmentation of the interferon response. So we thought, what if we could somehow cause alpha cells in our alpha ADAR knockout model to become in closer proximity to each other? And one way to do this is to increase the number of alpha cells in the perimeter of the islet. We did this by treating alpha ADAR knockout mice with glucagon receptor antagonists, which leads to alpha cell proliferation. And another way to change the islet architecture of these mice was to cause alpha cells to collapse into the core. And we did this by treating alpha ADAR knockout mice with streptozotocin, which leads to selective beta cell death, and in turn leads to alpha cell only islets. So I'll show you some results. This is an islet from an alpha ADAR knockout mouse where we see normal islet architecture and no inflammation whatsoever. We have GFP in green, glucagon in red, and CD45 in white. We treated wild type and alpha ADAR knockout mice with glucagon receptor antagonists. So this is a wild type islet, and we do see how there's a thicker layer of alpha cells in the perimeter of the islet. So more alpha cells, more communication between alpha cells. And surprise, when we treated alpha ADAR knockout mice with glucagon receptor antagonists, we found that this led to islet inflammation. Next, we treated wild-type islets and alpha ADAR knockout islets with streptozotocin, and we could see how, indeed, streptozotocin led to selective beta cell death. So we were left with alpha cell-only islets. And we were very excited to see that when we treated alpha ADAR knockout mice with streptozotocin, this also led to islet inflammation. So to summarize these findings, we suggest that in our ADAR deficient alpha cell model, we can augment the interferon response to levels that attract inflammatory cells by increasing the clustering of mutant alpha cells. And in this last slide, I'd like to explain how this all can fit into what we know about type 1 diabetes pathogenesis. So beginning with our beta ADAR knockout model. This model caused impaired RNA editing, which leads to the accumulation of endogenous beta cell double-stranded RNA, which is sensed by Th1, leads to interferon response, islet inflammation, and beta cell dysfunction and death. On the other hand, we have our alpha ADAR knockout model, which leads to the accumulation of alpha cell double-stranded RNA. This leads to sensing by Th1, but leads to only a minor interferon response. We think that maybe these differences between the models lies in first of all, differences in the metabolic load of the cell, whereby if we increase the metabolic activity of alpha cells, it may lead to transcription of more immunogenic double-stranded RNA. And our second hypothesis is that these differences stem from islet architecture, which limits mutant alpha cells from communicating and augmenting the interferon response. And now let's add some genetic factors that are known to be associated with type 1 diabetes into this model. So first of all, according to the paper that I mentioned at the beginning of this talk, it showed that RNA editing SNPs that could lead to deficient RNA editing. So these genetic variants could lead to similar effects that are shown by our model. Here we can see the variants in Th1 that are associated with type 1 diabetes. And maybe the addition of the well-established pro-type 1 diabetes HLA genes might push this model into autoimmunity and maybe full-blown type 1 diabetes. And what about the environmental trigger? We currently think that this trigger may be beta cell metabolic stress under this genetic background, which could ignite the type 1 interferon response that precedes type 1 diabetes. So I'd like to thank everybody at the Yuval lab, to Yuval and Agnes, my mentors at the lab. Thank you to Jonathan and Lisa, whose exciting new results I've shown today. A big thanks to all our collaborators, the Th1nk lab, Shalev Itzkovitz lab, the Al Powers lab, and to Ben Glaser and Klaus Kessner. Thank you for having me here, and I'll be happy to take any questions.
Thank you, Shani. That was really, really interesting, especially the unpublished data. Thank you for sharing that. And it is kind of interesting. We had, as I think I mentioned offline, we had Renee Van Tjoven from Bart Roop's lab in April talking about, sort of some of these RNA deficits leading sort of to the fragility of beta cells and delta cells. And so it's now, these stories are kind of, maybe moving in parallel. I'm going to ask, so I'm going to take a few questions. Yes, you can ask using the microphone, Afsan Arda. Hello?
Can you guys see me and hear me?
Yes, perfect.
Yes. Okay, great. First of all, I want to say congratulations, Shani. This is a lot of work, a lot of beautiful work, and very interesting stuff. And you did a great job with your presentation. It was really clear. So congratulations on that. So I have a few questions. It's mostly like curiosity. And maybe you said it and I might have missed it, but do you know the double-stranded RNAs that this ADAR is acting on? Like, have you guys investigated that in the cell metabolism paper? I can't remember. Maybe you mentioned it because it would be interesting to know, right? That um, if you can profile the double-stranded RNAs that the ADAR is acting on, that might be informative.
Yeah, definitely. Our collaborators at the Th1nk lab can see what transcripts undergo the most editing, and we have that information. And what we're trying to do now is really exactly what you're saying, is to maybe profile which double-stranded RNAs are in each cell and maybe eventually target them and show that, oh, this double-stranded RNA is immunogenic and this one is less immunogenic. So that's definitely the direction.
Great. And the other question I had was related to exocrine cells. I don't know if you know, but in the work from Kyle's lab, Kyle Galton, and my group, we've been looking at the genetic variants associated with type 1 diabetes risk, and the highest enrichment of these variants we see are in exocrine cells. So I was wondering, I mean, of course, this is like making another mouse model, but I think it would be interesting to maybe look at the effects. It's possible that there are like non-cell autonomous effects in acinar cells, let's say, right? When you remove this enzyme, that also triggers inflammation, right? And somehow, and if you're saying that the beta cells are more susceptible than alpha cells, that could be, that could lead to their death. But I think it's I think the based on the evidence that we see in humans, um, using for with the genetic variants, I think we need to consider the exocrine component as well. I don't know if you guys have considered it or it's in the plans.
Actually, we have created a mouse model that has adder deficiency in exocrine cells.
Great.
Yeah, and we're working on that. It
So
does lead to
look
inflammation.
great. Look forward to the results then.
Thank you.
Thank you.
That was such an interesting question and commentary, Efsan. I would say that I wonder, just getting back to the work you and Kyle Galton are doing, have you seen differences in these genetic variants in the exocrine cells? I mean, in the actual expression of the genes in the exocrine cells at different time points. So for instance, at um, early onset and later onset of type 1 diabetes, or is that not even a thing?
So I don't think the data we have is stratified as such. I believe that the GWAS is essentially the cohorts are just split. You have type 1 diabetes or you don't. But I think what you're saying would be interesting, like maybe to get to fine tune these susceptibility variants.
Yeah, I mean, it just
Like
seems like when you think about that kind of work and Shani's and Yuval's type of work, it's like a relapsing remitting disease, the pathology anyways, right? It shows up and maybe, especially in some people that maybe get it later, maybe 13 or 14. Seems like they may be relapsing and remitting. So then how might these mechanisms come into play? How might this ADAR piece come into play in someone who has this sort of like they have a little bit of, maybe they have a little DKA or something, then they recover, then they have a little DKA, then recover, then they don't, right? So it's just sort of a nice thought experiment to think about how these different mechanisms might be playing together across that timeline.
Definitely.
I wondered if Yuval has anything to say about that. He's in the call.
Val is silently listening to his speech. One maybe general comment that Shani did not mention because that was not her focus, but overall, what makes us very excited is that we see when you disrupt RNA editing, you see many phenotypes that are strikingly similar to features of human type 1 diabetes. And Efsan just mentioned the possibility that the exocrine pancreas might be involved. We haven't thought about that. We developed this model as Shani suggested, but we haven't pursued that in the context of type 1 diabetes. But for just one example of something that is making us puzzled is that ADAR deletion in young mice causes all the trouble that Shani described. But when we do that in older mice, not much happens, which is reminiscent of the mysterious severity of type 1 diabetes in young children versus adults. So we think there's more there for the comparison, and maybe there are common underlying principles that we could infer. But yeah, much more to discover.
Yeah, no, it's very interesting, and the time windows of these, the way these mechanisms are acting, and in context, I guess, is going to be very interesting as to elaborate on, which it seems like you guys are all working on that very big problem. So, Shani, this was just a really great, clear, very high quality talk. So thank you so much for walking us through your work and sharing, and we appreciate you taking the time.
Thank you so much, Monica.
Get a
It's
lot
great
of
to have you.
accolades in your chat. So congratulations, and we'll keep an eye on what you've got going next. Have a great rest of your evening.
Thanks, you too.